Hello, this is our active transport video, so let's get to it. Active transport is movement of particles across a membrane from an area of low concentration to high concentration. So unlike diffusion, osmosis, and facilitated diffusion, this time we are going against the concentration gradient, and this requires energy in the form of ATP. There are many transport proteins involved in this, but we're going to review three, and those three transport proteins are uniporters, symporters, and antiporters. They have some similarities between them. Uniporters move one particle against the concentration gradient across the membrane, and they can move only in one direction. So if this uniporter can move from outside of the cell to inside of the cell, it will never be used to move a particle from inside the cell to outside of the cell, and it's moving against the concentration gradient. They work the same way that carriers work. First, they attract their particle via polarity. That triggers an isomerization, which moves the particle across the membrane. Because of the isomerization, the polarity of that region is different, and the particle is no longer attracted, so that it is released, and then the protein renatures. So, same steps as carriers. What is different is that they require ATP. So during the isomerization or the renaturing step, ATP will lose its phosphate and become ADP. The phosphate will bind with the uniporter, and its charge will disrupt hydrogen bonds and cause the uniporter to change its shape. This is called a conformational change because it's a generic shape change for the protein. So maybe it's for the isomerization step, maybe it's for the renaturing step, but that's caused by the phosphate donated by ATP. Symporters are slightly different than antiporters. They carry two distinct particles, and they always move them together in the same direction, but one of those particles will be going with the concentration gradient, and one of those particles will be going against its concentration gradient. They function the same way that uniporters do, with the same steps: attract, isomerize, release, renature, and they do use the ATP in the same way as well. There are some variations, however. Symporters can function in one of two ways. They can grab all of their particles at once and renature and isomerize and release them, and so they can move all the particles at once, or they can move their particles in sequence, where one of them is attracted and it renatures, or it isomerizes, which allows the other one to be attracted, and then it renatures, and then the first one is attracted and it isomerizes, and the second one is attracted and it renatures. So they can move all at once or in sequence. Another thing about symporters is that they can move all their particles. They can move particles in groups. So it's not that they only move one of particle A and one of particle B. They have variable numbers, so they could have three of particle A and one of particle B, and that will be determined by each symport. So there's a unique ratio for each symport that they move. And lastly, if they are moving in sequence, then ATP may be used to move the particle that is moving against its concentration gradient. Finally, we have antiporters, which are very similar to symporters. They move two particles, and one is with the concentration gradient and one against, but this time they're moving in opposite directions. They can move their particles in groups, so they can have a different ratio than 1 to 1. They can move 3 of one and 1 of the other, or 2 of one and 3 of the other. So they can move multiple particles in groups. But they will be moving their particles in sequence. So they'll move 3 of particle A and 2 of particle B, and then 3 of particle A and 2 of particle B. And because they're moving in sequence, ATP may be used to move the particle that is moving against the concentration gradient. So let's review. Active transport is moving particles against the concentration gradient with the use of energy in the form of ATP, and it involves transport proteins. Uniporters are one protein that move one specific type of particle against the concentration gradient. They are one-way motion, so they only move them one way, and a different carrier might move a particle in a different direction, but that carrier will always move them in the same direction. They have the same functional steps as carriers. They attract the particle, they isomerize and move the particle, they release the particle, and they renature. And either isomerization or renaturing will require ATP. ATP will lose its phosphate and become ADP. The protein will bond to the phosphate, and its charges will force a conformational change in the protein. Symporters move two distinct particles in the same direction, one with the concentration gradient and one against it. They function and use ATP the same as uniporters, and some porters can move their particles in groups instead of one at a time. And some move their particles all at once, and others move their two different particles in sequence. And when they are moving in sequence, ATP is needed to move the particles that are going against the concentration gradient. Finally, antiporters move two distinct particles in opposite directions, one with the concentration gradient and one against. They function and use ATP as uniporters do, and some may move their particles in groups, but they generally move their particles in sequence and use ATP to move the particle that is moving against the concentration gradient. I hope this helps. Struggle well.